As you all know, this is the Nobel Prize season, and of course, it becomes imperative for me to discuss some things about the Nobel Prize. Not all the Nobel Prizes, of course, it will take a considerable amount of time, but one interesting Nobel Prize we'll be discussing because it deals with medicine, physiology, and will be the agenda for today will be to discuss some of the facts, some of the history of the Nobel Prize, one important Nobel Prize which is of peculiar interest. It will take us back to school, deals with DNA, genes, chromosomes. So I also had to revise a bit the concepts. So you will enjoy this lecture. I'll make it as simple as possible. And finally, we'll conclude with the relevance of the Nobel Prize today. Okay, so Alfred Nobel. So Alfred Nobel. So who was he? Okay, so he was a Swedish chemist, engineer, and industrialist, credited with the discovery of dynamite and other such explosives. So not very noble, Noble. Anyways, so it is so in his will, his demise took place in 1896. But in 1895, in his will, he said that whatever fortunes he had earned, whatever money he had earned towards his lifetime, in his will, he mentioned that should be given in the form of prizes that should be used to constitute such prizes, and that should be given to those people previous year, who in the previous year or the preceding year have made the greatest contribution to mankind. Fine. So, as per his will, as per his wishes, a Nobel Trust Foundation was created. Nobel Foundation was created, and which is responsible for administrating those funds. Okay. So, this is the history of the Nobel Prize. And basically, you might have heard that those hardworking, dedicated scientists or physicists or those who have worked towards the world peace, they are called Nobel laureates. So this laureates, again, interesting fact, it is derived from the Greek word laurel. So laurel wreaths were presented to victors. The victors in ancient Greece were honored with the laurel wreaths, this leaf-like structure, round leaf-like structure. So this is the history behind why the Nobel winners are called Nobel laureates. Fine. Okay. So according to his will, so five five Nobel prizes were constituted. Okay. So physics, chemistry, medicine, or physiology, peace, and literature. And these awards were to be presented by different institutes. For example, physics, chemistry, they are to be conferred by the Royal Swedish Academy of Sciences. Fine. This medicine physiology prize is conferred by the Karolinska Institute, again Sweden. Okay, Nobel Peace Prize is conferred by the Norwegian Nobel Committee. Okay, and this for literature, we have the Swedish Academy of Scientists. So these two are different. So these four institutes were given. Now, what happened? So the first Nobel Prize was conferred in 1901, 5 years after the demise of Alfred Nobel. And in 1968, another prize was constituted. So it was constituted by the it was constituted by the Bank of Sweden, and it came to be known as the Nobel Prize in Economics. And this Nobel Prize for Economics is also conferred by the Royal Swedish Academy of Sciences. Fine, so this is the history of the Nobel Prizes. Fine, so these basics are clear. So it was this economics Nobel Prize was constituted in 1968. It was first presented in 1969. Then, so this just to make it a little bit interesting, the mapping. So this is the map of Europe. So Norway and Sweden. So Norwegian Nobel Committee is responsible for conferring the Nobel Peace Prize, and Sweden for the other prizes. Fine. Then other interesting prizes. Now you might have noticed that other subjects, sciences, a lot of them are missing. So I happened to come some I had read during my preparation years for the competitive examination. So I remembered some. So you might have heard about this prize, Pritzker Prize. So what it is for? It is for excellence in architecture. Okay, it is not a Nobel Prize. Then there is the Fields Medal. You might have heard of Fields Medal. So this is a prize given for excellence in mathematics. Again, not a Nobel Prize, but considered equivalent to Nobel Prize. Another prize given in the fields of mathematics is the Abel Prize. Okay, so just for general knowledge, it is interesting. Then I came across another prize recently, this Kavli Prize. Kavli Prize. So this is given to people with excellence or contribution in the fields of astrophysics, neuroscience, and nanoscience. So it's good to have general knowledge. Okay. So we are going to discuss today the Nobel Prize in the field of medicine or physiology because it holds immense implications and applications for curing diseases like cancer, diabetes, and various autoimmune disorders. Okay, so these have been conferred to two persons, Victor Ambrose and Gary Rufkun. But in order to understand what exactly these persons did, we need to go back to school a bit. Okay. So, first of all, what I want to discuss is the basics of heredity and evolution, DNA, genes, and chromosomes. So, let's start with the in simple words. What is DNA? Full form is deoxyribonucleic acid. So, it is a kind of instruction manual. In simple terms, instruction manual, and it defines the structure and the building of an individual. Okay. Then it has a double helix structure. So this is about DNA. Now, what is a gene? So before that, some other characteristics. So in DNA, what we have are various bases. So bases, we have four major bases that are adenine, cytosine, guanine, and thymine. Fine. So if you remember the structure, this is a double helix structure which represents the DNA. Fine. So, okay. So this stair-like structure, this nucleotide structure, okay, this is made up of basically these are the bases. So at one end you have adenine which pairs with thymine, and at other pairing you have cytosine which pairs with guanine, and the rails, these are the bases which form the steps of this helical staircase, and these handrails, they are made of phosphate and sugar molecules. Okay, so this is like an instruction manual. For example, in any computer language we have some words. So in words, so these bases act as letters or alphabets, combining which we can prepare an instruction manual. So that instruction manual is given in the form of genes. So next, after DNA we have genes. So what are these? These are the building blocks of any organism. Fine. These are a set of DNA. For example, a DNA is a very long helical structure. If we pick some of the, for example, consider this DNA as a toolbox. Now, these genes are responsible for exhibiting some physical characteristics, some proteins. For example, they will define the hair color, the color of eyes. Okay. And this DNA, some molecules like muscle cells, nerve cells, they require to activate a particular set of DNA in the form of genes to facilitate their appropriate function. So if we So if we take, suppose this set of genes is responsible, this set of DNA is responsible for a particular formation of genes which exhibits the hair color. So this will be the function of genes. Fine. And finally, when we come to chromosomes, what are chromosomes? These are the thread-like structures present in the nucleus of the cells, and these contain both DNA and genes. Obviously, genes are nothing but groups or proper combination of tools of DNA, and this actually defines all the instruction. This collates and supervises the instructions which are to be followed for the functioning of various molecules in the human body. Fine. To make it simpler, this diagram I have fixed here. So here you can see, so this is the cell. This in the middle we have nucleus. Nucleus has chromosomes. Chromosomes have these double helical structure known as deoxyribonucleic acid. DNA is composed of the stairs, which are composed synonymous to stairs, which are the base pairs, adenine, cytosine, guanine, and thymine. And the side structures, they are made of sugar and phosphate molecules. And these these segments, like this purple colored or this segment, depending on the particular instruction manual or instruction set that is to be used by the molecule or the cell, this corresponds to a gene. Fine. So chromosomes, we know that there are 20 in humans. There are 23 pairs of chromosomes, out of which one pair is belongs to the sex chromosome. Fine. Now we have to discuss what's important for this discussion in the Nobel Prize, the microRNA. Before discussing microRNA, we need to understand what is RNA. So RNA stands for ribonucleic acid. So what is RNA? RNA is made from DNA by a process known as transcription. This is very important. Fine. So what happens if this is, or to make it simple, if this is the DNA structure, so for the formation of RNA, there is an enzyme known as RNA polymerase. So this RNA polymerase, what it will do is, it will take one staircase part or one segment of the DNA, and with the help of this polymerase, RNA polymerase, this will help in unwinding or taking one set of DNA which will constitute a gene, and it will sort of form a template to form a type of RNA known as messenger RNA. The basic function of RNA is it is a functional part of DNA. So what RNA does is it helps in creating a template of genes or template of instructions, because DNA only contains the information, and that information is to be used via the various cells. For example, we have muscle cells, we have nerve cells. So for that, we need to activate, those cells need to activate a specific portion of DNA, which is known as gene. And those synthesis of those genes, which are expressed in the form of proteins, those making of proteins which will exhibit or perform those particular functions, that will be done by RNA. So that RNA, with the help of an enzyme RNA polymerase, it will take a template or blueprint or a design from the DNA and then further give other molecules or other enzymes the platform to use this blueprint to form proteins. Fine. So this is the basic concept of RNA. So RNA is the functional part of DNA. Don't worry, I'll show you the images also to make it even more clear. So like you can see in this picture, so you can see in this picture, so this is a molecule. Okay, it has chromosomes. Chromosomes are made up of DNA and genes. So here we can see different sets of tools. DNA is a set of tools, alphabets. Different alphabets or tools, they are sets and used as genes. Okay, so for example, here we have, say, nerve cell. So the nerve cells, they don't require the whole DNA. They, in order, nerve cells function is to send signals to and fro from and to the brain. And for that, they need to, the nerve cell molecule, it needs to activate a certain set of DNA known as a particular gene while switching off all other genes. So this gene, in order, so who will tell that nerve cell that you have to use only this set of DNA, only this gene to perform your function? So that is given by the messenger RNA. Fine. So, and messenger RNA is formed by the template, by from the functional part of DNA, when the DNA forms a template. So this process is known as transcription. And finally, protein. So what is the protein? Protein is the basic, it is the, for example, transcription, it does just gives a blueprint or a template to RNA. But now that blueprint cannot perform the functions of any cell, whether it be nerve cell, whether it be muscle cell, to, it will not perform the functions on its own. It requires proteins, it requires molecules to perform those functions. So that template, that blueprint, for example, we have to make a building and we have a plan. So that plan is given by mRNA, and then mRNA is then bound by other molecules for protein synthesis. We have the basic constituents of protein, which are known as amino acids. So amino acids are then formed into, they are converted into proteins. And then this nerve cells, for example, if there is a muscle cell, so this muscle cell has this entire DNA, every cell has DNA, every cell has genes. But in order for this muscle cell to do its function, for the contraction, dilation, activation of muscle cell, it should be given such an instruction that it doesn't use this gene, it doesn't use this gene, it only activates this gene. So that template is given by mRNA. mRNA then binds with other molecules to form protein by the process known as translation. And finally, the cell comes to recognize that only this gene has to be activated when I have to perform my function, the function that has been allocated to me to perform this function properly. Fine. Now, the important another term, gene regulation. So what is this? This we have been talking right before this. So gene regulation is the ability of a cell to activate a particular set of DNA, or in other words, genes and switching off other genes. So this is known as gene regulation. Fine. Now what happens is we had talked that the microRNA for which Mr. Rufkun and Mr. Ambrose have been conferred with the Nobel Prize in Physiology or Medicine. So they have come up with another concept. They have discovered microRNA. Till now, we were only aware of messenger RNA, which is the functional part of DNA formed during the process of transcription. Okay, now what their discovery was, so just to again emphasize or elaborate upon this process. So here we have DNA. The functional part of DNA, this is RNA polymerase. It is RNA polymerase by the process of transcription. It helps in the form of formation of mRNA, which gives the blueprint for the formation of proteins, and it in combination with other molecules, it leads to the formation of polypeptide chains, which are nothing but chains of amino acids which are responsible for the formation of proteins, and these proteins allow the particular category or particular type of cell to perform their required function. Okay, so how did this microRNA take into place? First of all, if we want to define microRNA, how do we define? So this we have already discussed, the mRNA or a messenger RNA. So in order to define microRNA, this is defined as that part of RNA or that part of messenger RNA, small part, which latches onto this mRNA and inhibits the protein synthesis. For example, now we are coming to the basic what we are trying to achieve. So sometimes what happens is we have understood what is gene regulation. So sometimes when gene regulation or the appropriate switching on and off the genes does not take place normally, it is led that the it leads to the abnormal gene expressions. The cells don't behave normally or as they are supposed to. So, this expresses it in the form of diseases such as cancer, autoimmune disorders, diabetes. Okay. Now, so in this microRNA is related to this. Now, what these two guys did, they studied this worm. This is 1 millimeter long worm. So, this is known as C. elegans. Okay. So they have various genes which are similar to those genes present in tissues in other large mammals. So they were of particular interest. So these two people, Mr. Rufkon and Ambrose, were interested in two genes, LIN4 and LIN14. So they observed that this LIN4 is inhibiting the functioning of LIN14. These are genes. So, for example, if LIN14 is supposed to form a protein for the maturing of a particular cell in this worm, so this LIN4 is not allowing it to do so. So, they both started thinking that what might be the actual reason. So, from that, they came to the discovery of microRNA. How? So, for example, if this is a set of DNA which corresponds to the LIN14 gene. This is a LIN14 gene, fine. And this is first getting transcripted to form the messenger RNA. This is getting formed the messenger RNA, and this messenger RNA in combination with ribosome or other molecules, it is leading to the formation of the necessary or relevant protein. But what is happening in this case of the C. elegans is that this Lin 4, Lin 4, the the RNA, this Lin 4 gene is also producing its RNA and subsequently messenger RNA, but this is also producing its own microRNA, which resembles a small part of the messenger RNA of this Lin 14, and it is latching on to this messenger RNA of Lin 14, and thereby it is inhibiting, it is not allowing that protein to form. So, how is it now applicable in medical science? So, if we have to cure some disease or prevent some disease, so if, for example, there is an unregulated gene which is causing the production of cancer cells, so that is due to some protein is there which is leading to the formation of the cancerous cells. So, if we somehow inhibit the formation of protein so that cancer cell will not be formed, that diabetic or autoimmune disorder causing cell will not be formed. So if we create some kind of RNA to inhibit the function of protein formation by this messenger RNA, we can cure this disease, and that is why this discovery is very essential. So I hope you get the gist. I'll show you this. So this microRNA, so here also just repetition, we have shown that it holds great promise implications in the fields of medical science. So this is a great leap forward. So now, so this was all about the Nobel Prize in medicine. Now the last point is relevance. We have to discuss that is the Nobel Prize relevant still today? So now we know that whenever the Nobel Prizes are declared or they are conferred, a lot of journalists they rush to stake claim, put it in the news and all that. And now, not only just discuss who has won the Nobel Prize, but what is the application, what actually the scientists, the physicists, or the chemists have done to. So that is important. For example, it also, when novel discoveries are made, the STEM sciences have been popularized in countries, developing countries like India. So, science, technology, engineering, mathematics. So, it inspires interest. But what happens is because there is too much interest involved, too much research involved, it is not necessarily bound to evoke that kind of interest to enroll the students in the STEM sciences. Fine, but nonetheless, it gives an idea. It's a period of excitement. It gives an aspiration, inspiration to the various students or various aspirants all over the world to get into this field of STEMs. Now, another thing is the myth of the genius scientist. Now, obviously, whenever some discovery is made, it is made by a group of scientists, not by just by single or one or two scientists. There are a lot of collaborators. So they don't get the appropriate recognition that they deserve. Okay, so this is again. So then this research organization or this Nobel is somewhat inclined or has a propensity towards the Western countries. Over the period of which the Nobel Prizes have been granted, so majority of the prizes have gone to countries such as US, UK, Germany. So what I mean to say is that it has been observed that the developing countries or the countries which possess low infrastructure, low research facilities, they are being overlooked. When we consider India, countries like China, they have received very less amount of Nobel prizes. Okay, and moreover, it has also been criticized of sexism. Means they are being criticized of being biased against women. Only around 15% of the women Nobel Prize winners till now have been women. Out of those, only one woman has won the Nobel Prize twice, that is Madame Marie Curie, radioactivity. So, this myth of these genius scientists, then, so these are some of the drawbacks or flaws which exist. So, what we have to go, we have to look forward, we have to, the Nobel committees, the Nobel Trust have to look forward is to share all the expertise, the technological information with the countries to help them progress forward also. So this was about only one Nobel Prize, so it took a significant amount of time. Other prizes you can read or I will discuss if I have time in the future. So till we meet again next time, Jai Hind.